From Microbe TV, this is Office Hours for Wednesday, May 8th, 2024. I'm Vincent Racaniello, and welcome to your viral corner of the internet. Welcome, everyone. Good to see everyone. I'm here at the incubator in New York City. Karen is not here. She's on the road doing some recordings with the immune team. And I want to start off tonight. I'm going to ask you where you're from so you can start queuing up your answers. Let's see who's here tonight. Well, let me thank our moderators. First, we have Andrew from New Zealand. Can't make it today. Stuck in a meeting. Okay. Sorry, Andrew. Steph SF is here. Vanity Nutrition can't make it. She's got an exam tomorrow. Thanks for popping in. Les is here. So basically, we have Les and Steph to hold down the fort. We don't have any of our other intrepid moderators, but that's fine. We'll be fine. And there are so far 63 of you. Don't forget to hit the like button to try and attract other people, to teach them some science. There is, you can support us if you like our work. We'd love to have your support. We're growing. We need support to grow, and you can see above my head there's, is it the right finger? Yeah. Our microbe.tv/contribute, or, wow, I went in the right direction. Look at that. Or Venmo, or if you go below the screen, which would be, let's see, where in the screen is it going to be? It's underneath the text. And I'm not real time here, so I can't. And now I'm looking at myself. I decreased the latency tonight. Yeah, so this is right. This is kind of near where the text box is. Underneath it, there's a $. It's called the Super Chat. You can give money. Now, YouTube takes a cut of that, where everybody takes a cut, right? It's all about the VIG. But whatever your pleasure, I would appreciate it. Let's see where you're from tonight. Office Hours, Brad Badcat is from Winthrop, Maine. So here in New York City, by the way, it's a nice day. It was right now, wow, in Chelsea, which is the neighborhood that the incubator is in. We're right on the cusp of Chelsea. It's 26 Celsius, which is something like 82, right? And I don't know what, there's no window here, but it's dark, so it doesn't really matter. So it was a nice day, but I was here inside all day, and I don't know what was going on. So Bad Cat is from Maine, where it's 47. Wow, that's different. Tricia is from Massachusetts. Marie is from Dolton, Illinois. Rima is from Iowa. I forget all the time. In 2 states, actually. Super chat from Kip and Laura. Thank you. So that's a super chat, folks, which is how you can give some money right here at YouTube. Thank you for your support of science communication, Kip. Really appreciate it. No guest today. Yeah, I know it's a few weeks in a row without a guest. I'll try and get one next week. So my course is now over, so that's a big chunk of time back. They're taking the final exam this week, actually, at their leisure, because it couldn't be scheduled on one day. They have to have flexibility, given all the issues that happened. Wayne is from Urbana, Illinois, where it is 70 F. That's a good temperature. And then on the other side of the world, Dagfinn is from Oslo. Got to make it to Norway one day. I know there's good virology there. I have to hit all the countries where there's good virology. That would be cool. Do podcasts. Luis is from Guatemala, where we used to have 22 to 25, no more. I guess it's no longer like mild, right? It's really hot. Sorry about that. Will is from Changsha, China, where it's 22C. Pete is in London. Mark is in Tacoma, Washington. Tacoma, Washington. Hello, Mark. I'll get back to you about my thoughts on Novavax. Zandanga is from Florida, 81F. Simon is from San Jose. Mark says, I have to take a time point, do some dilutions and plating. So I know exactly what he means. Maybe not all of you do. He's doing a timed experiment where something is happening over time, and he's taking samples from whatever it is. It's a culture most likely, but it could be something else. And so that's a time point. And then he's making dilutions and plating it out. So maybe he could be plating bacteria. He could be plating phage on lawns of bacteria. I don't know. Don't know what he's doing, but that's the idea there. I love it. Jessica is from Toledo, Ohio. Lori is from Charlottesville, Virginia. Some good questions. Lise is in New York City. 2 flight delays, huh? Too bad. I'm sorry. Lise is visiting the incubator tomorrow. No, Friday. Sorry, tomorrow is Thursday. I need another day this week to get stuff done. Actually, I have a lot of visitors tomorrow from my course. A bunch of people throughout the day are visiting. Bernard is on a train to Chicago. Claire is from the UK. Now, of course, many of these names are familiar, but there are always new names, which I love. And so, for example, Theodore is from Corinthos, Greece. Cool. That's very cool. You should be around when I have Theodore Hatsiwanu as our guest in a couple of weeks. Angela O'Kelly is from Ireland. This name I don't recognize. I think it's cool. O'Kelly. Isn't that great? Jonathan from Encinitas, California. Kang is from Chicago. 19C. Partly cloudy. Mr. Doreen is from Elgin, Illinois, sitting outside. Yes, it's not 2C. It's 22C, right. Leo is in the Mendocino coast. Kang's got a question. We'll get to it. Joseph is in southern Ontario. Christopher is in Montreal. Really appreciate all I've learned from you and your team. Thank you. I'm glad you learned. It's our goal is to teach you and make it as effortless as possible, right? By the way, I'm going to have a mini lecture for you tonight about prions. It'll be fun. I love prions. I don't want to have them, but I love the concept. James is in Longmont, Colorado. University of Toledo representing. That's right. EHS Talent is in Eastern Pennsylvania. We'll take good care of you if you come to Oslo. I'm sure. What is the word now? I'm missing because I don't say it the hospitality of the Norwegians. I would love to. Unfortunately, I'm going to Sweden and Finland in a couple of weeks, but I don't have time to get to Norway. But I'll be back. I love to travel and do podcasts. I don't want to just travel for its own sake. It's not so much fun. But to do some science, do a podcast, go to a meeting, and then look around, that would be fun. But I usually don't do that. Grace Rosella is from Melbourne. Wow, that's far away. One of the farthest. And we have Visto from Sydney. 2 people from Australia tonight. Very cool. Genetics is in Phoenix. Famida is in Chapel Hill. And this is rattlesnake weather. Really hot weather, huh? Wayne broke the chat out to a separate window. Now I don't see a thumbs up icon. I don't know anything about that because I don't look at I have the chat in one window. I don't break it out. But maybe someone could help. Or put the window back. Stephanie is from Mexico. Wow. That's cool. Yes, I'm live. I'm right here live. Just at work today at the incubator. I had my dinner. I did a bunch of editing today and posting. If some of the people were missing some of the recent Tweevo episodes, I just posted some of them, but not all of them. I still have 2 to go. Good productive day. Rima is in Iowa. I got it right. Iowa. There you go. All right, Wayne found it. Good, good. Abdul is from Saudi Arabia, where it's probably really hot. Another place I should go. I know there's virology there. One of these days. John, of course, is in Minneapolis. Up at the cabin to put the dock in today. I look forward to watching when I return. Have a great office hours. Okay, John, thank you for your support. I'm sorry you won't be here. You are always a good part of the chat. Krishnan is from Malaysia. This is a great audience tonight. You know, when we sign off, you all have to say goodbye in your native language. Privyet, everyone. I learned last time Privyet and Dobry večer. You know, kind of like good evening and hello. It's great, but it's Russian. We have to get some other languages in there. Adriana is here in New York City. Adriana Aquino. Cool. Aquino. I don't remember. Zayn is in Australia. 3rd Aussie tonight. That's cool. That's very cool. Steve is in Columbus, Ohio. Columbus also is well represented. Ohio is well represented here on Office Hours. TB is in Poland, Warsaw. I don't know how to say it properly. 1963, they had a variola outbreak. Frank is from Santa Barbara. Mark says, I need to come to visit the incubator this summer. You need to show me the best place to get a slice of pizza or other New York City foods. Yeah, I don't know anything. I don't know the best of anything, but we'll figure it out. We'll ask Amy. She'll know where the best of everything is. I don't know if Amy's watching, but you'll tell me where to take Mark. David is from Dayton, Ohio. Cheryl is from Santa Rosa, California. Oh, there you go. No, Cheryl, I just got your lovely little letter, lovely handwriting, and now I could put a face with that writing. You look like you're writing. There you go. Thank you so much for your support and your kind words. I don't think I have it here. We hung it up on the board. We have a board for love notes, and it's up there with others. Thank you so much. You can write us a letter here at the incubator. The address is on the website in the contribute button area. Our favorite nephrologist, Carol, from Southern California, where it's sunny and warm. Oh, and look, one of our other moderators, Peak, who has not been around for a while. Welcome back. Hello, currently in the lab, casting a gel and coating an ELISA plate. I have some time while I wait for my resolving gel to harden. Very good. Good to see that you're doing experiments. Ricky is from the bourbon capital of the world, Bardstown, Kentucky. Oh, well, I wouldn't have known that. Mike and Lori are from Seattle. Welcome. And Veronika, privyet, and dobry večer. Finally, I got someone else to practice it with. Eliza is from Southern Ontario. Intersoln is in Northern Ireland. It's 54F, but no one who started school after 71 uses Fahrenheit. Still not an heir as those who began their education before 1970 would say. Well, I did. I began mine in the 60s. There you go. Oh, Tom is here. Welcome, our moderator, Tom, from the Oregon Coast Range. Very nice. Niall is from Colorado. Karen's playlist is in northern Maine. Questella is in Ohio. See, some of you, I can fill in where you're from because I've seen you here so long. It's great. Lucy is from Sechelt, BC, British Columbia. Moisha is from New Orleans. Cool. Well, I was there not too long ago. What year was I there? Yeah, I've been there a couple of times in the last couple of years. Yep. And Robin is from Sultan, Washington. Welcome, everyone. Good to see you all. And I will get to your questions, but I would like to give you a little mini lecture on prions. By the way, Friday on TWIV, we have a guest, Richard Webby from St. Jude's Children's Hospital, who is an expert on influenza viruses. He's going to talk about H5N1. You won't want to miss it. And there's some more people who have just showed up. Let's see. Spamlet is from Bedfordshire. And Martha is from Charleston, South Carolina, also the home of Michael Schmidt. All right, let's do a mini lecture. Let's see now. I have to cue this up, get it working. I can do this. And I think I did it full screen last time, right? No, but I couldn't because I wasn't able to select the side. So first, let's turn on my camera so you can see me, and then we'll do a screen share. And that should do it. She'll make this big, get rid of this format. And I'm just going to wait a second to see it in my monitor here. It looks pretty good. It's not cut off, right? Tell me if it's cut off, folks. Nobody's saying that it's cut off, is it? It doesn't look cut off to me, so we'll go. So this is lecture 24, the penultimate lecture in my virology course. That means the second to the last, right? And thank you, everyone. Not cut off. In this lecture, we address some interesting questions like, what's the minimum genome size needed to sustain an infectious agent? And could an infectious agent exist without any genome, right? Without any DNA or RNA? And this lecture, we talk about viroids, satellites, and prions to address these questions. So today we're not going to talk about viroids and satellites, but viroids don't encode any protein, and they're tiny, 100 bases, 120 bases of RNA. Satellites may not code in any protein either. And prions are just proteins, no nucleic acid, and they're infectious. So the answer is really small, and yes, you could exist without any genome. So what is a prion? Prions are infectious proteins, no nucleic acid. I found this street sign years ago, Prion Road. Would you like to live on Prion Road? This is funny. You know why? Prion is a word made up by Stanley Prusiner. He, take infectious protein, and if you read his biography, he was having a tuna fish sandwich one day at the UCSF, and he had this idea to call it the infectious protein, PR, prion, and then it didn't sound right, so he added O, and it's prion. And now I don't know where this Prion Road came from, because maybe prion is a word before, right? And maybe they copied it. I don't know. So, but anyway, it's a funny picture. Anyway, they're always in the news, right? BSE, mad cow disease, bovine spongiform encephalopathy, Creutzfeldt-Jakob, scrapie, kuru, chronic wasting disease of deer and elk. We'll talk about all these today. And in 97, Stanley Prusiner got the Nobel Prize for figuring out that these are infectious proteins. Now, you may have known about George Balanchine. He was the director of the New York City Ballet. Used to go to the ballet a lot when I lived in the city, and he died of a prion disease. And this is an article from the Times about how they post-mortem took pieces of his brain, and they saw that he had the typical pathology of a scrapie disease, and he developed it insidiously. He started having trouble walking. He used to wear these jackets with the leather on the elbows. You remember those? They're still around, but they were red leather, and people noticed that the walls to his office started to have red streaks because he was bumping into the walls. He couldn't walk properly, and he got worse and worse. He lost motor function, cognitive function, and he died. And it turned out he had a prion disease. So these are called transmissible spongiform encephalopathies. All right. So an encephalopathy is a disease of the brain. It could be anything. And these are all fatal neurodegenerative disorders of mammals. As far as we know, non-mammals don't get them. Fatal, 100% fatal. Nothing you can do about it. There's no treatment. And thousands of people are diagnosed every year globally with TSEs. About 1% of those arise by infection. 99% are either genetic or spontaneous. And we know that at least 120 people got a variant form of Creutzfeldt-Jakob from eating meat from cows who had BSE, and we didn't know it, and it got into the food chain. Yes, Mark, most vertebrates have PRPC, but only mammals get, as far as we know, only mammals get prion diseases. So there are many different diseases of animals. These are all mammals, right? BSE in cows, chronic wasting disease in cervids, exotic ungulate encephalopathy in Nyala and greater kudu. Yeah, those are species I see all the time. Feline spongiform. You get cats, your kitty cats and great cats get it. Sheep and goats get scrapie. And there's transmissible mink encephalopathy. The TSEs of humans are here. Creutzfeldt-Jakob, fatal familial insomnia, Gerstmann-Straussler, Kuru, and variant CJD. The last one is what you would get if you ate contaminated cow meat in the 90s in the UK. We'll talk about that. Spongiform, why are they called so transmissible? Because, about 1% of them arise by transmission. Spongiform, the brain has sponge-like holes throughout it. This is a section of sheep brain. It's not supposed to have these holes. It's supposed to be solid, and this is bad. It causes psychomotor dysfunction. It causes cognitive issues. And the symptoms for these different diseases that I just mentioned here, they're all slightly different because the prions are misfolding in different parts of the brain. The story begins with sheep. The first TSE, transmissible spongiform encephalopathy, the first recognized. The infected sheep rub on fences, right, and they scrape their fur off. These sheep get motor disturbances. They get uncontrollable trembling, which was called tremblement de mouton in Canada when it was first observed. Paralysis, weight loss, death within 4 to 6 weeks after the onset of symptoms. And it's been known in European sheep for over 250 years. It's endemic in many countries. In the UK, still 1% of sheep every year come down with scrapie. It just happens spontaneously in them. Now, the farmers started to figure it out. They realized that if they brought a sheep to another farm that had scrapie, why would you do that? You give your neighbor a sick sheep. Then the neighbor's sheep could get it. So it's an infectious agent. And people started to work on this. And in 1939, they showed that the infectivity in sheep brains can pass through filters which pass only viruses. And so for a while, people thought, it must be viruses. But they're highly resistant to ultraviolet light, ionizing radiation, and formaldehyde, all of which would inactivate viruses if you give them enough. And so the idea came about that these agents don't have nucleic acids in them because they're so resistant to UV and ionizing radiation. So they're not typical infectious agents. They are unusual infectious agents. And this was an early experiment done in 1984 that shows how resistant prions are to ionizing radiation. So this is radiation in increasing doses here. We're giving a ton of radiation. We're radiating different viruses here, and then scrapie. So scrapie, you need the highest dose to inactivate it. And then As you know, once every 2 weeks, the board just dies, and they turn it off and on. So these on the top are DNA viruses. So the bigger DNA viruses with big genomes, they don't need much ionizing radiation because their genomes are a big target. And as you get smaller and smaller, you need more and more irradiation to inactivate. But scrapie is just bigger than everybody. And then at the bottom here, we have RNA viruses, which follow the same trend. So people began to think that there was no nucleic acid in these. And I have to remind you, you don't know you have a TSE until you have symptoms. And then it's too late. You can't do anything about it. It would be too late even before, if you picked it up before symptoms, which you couldn't do in theory. But they're all invariably fatal. They progress to death in every case. So very scary. Now, in 1967, a mathematician said these TSE agents were proteins. And he had looked at the data and decided that. In 1981, Stan Prusiner purified from infected sheep brain, from scrapie brain, a protein which, when injected into animals, would give them the same disease. So he called it a proteinaceous and infectious particle. So you see, proin was the name he first came up with, proin, but he didn't like the sound of it. So he changed the letters around to make it prion. He swapped the I and the O. The genesis of a made-up word. And the protein is encoded by a gene called PRNP, which is essential for the pathogenesis. If you take mice and knock out the PRNP gene, they're never going to get prion diseases. They're never going to get TSEs. So our current view of how this works is the pathogenic prion is a conformational isoform of a normal host protein called PRPC. This is what Mark Martin just referred to. Prion protein cellular, C stands for cellular. It's the normal form, and the pathogenic form is called PRPSC in honor of scrapie, but it applies to any of the pathogenic forms, is misfolded and causes problems. This PRPC is on the outer surface of neurons, and when you introduce the abnormal conformer, which is a name for the abnormal conformation of the protein, PRPSC, when you introduce it into the organism, it causes the normal PRPC to misfold into the pathogenic conformation. Isn't that insidious? So you have all PRPC in you, and if you get a little bit of PRPSC, the misfolded version, it causes all the PRPC to misfold. And eventually that accumulates and you get your TSE, and then you die. So here's how the conversion looks, PRPC to PRPSC. So PRPC protein has a couple of alpha helices and 2 beta strands. And the PRPSC, the misfolded protein, has a lot of beta strands here. You can see them, they're diagrammed, these little beta strands forming beta sheets. And so this changes many properties of the protein, including its susceptibility to proteinases. And that's an assay for differentiating the two proteins. So if you digest PRPC with an enzyme called proteinase K, which will cut it, it cuts the N terminus off, and you get a 27 to 30,000 Dalton fragment left. Sorry, that's the PRP SC. The PRPC, when cleaved with protease K, is completely digested. And so you can see if you've got misfolded conformers, PRPSC, by doing a digestion and then assaying for the integrity of the protein. So just this conformational change makes it very resistant to protease digestion. Now, one of the important experiments that led to the prion hypothesis, our understanding, is that mice lacking both copies of the gene that encodes the prion protein are resistant to infection. And so what do I mean by infection? Well, you can take sheep prions and inject them into mice. And if the mice do not have a prion gene, which they can live without, they never develop a prion. So you can introduce PRPSC protein into experimental animals by injecting it. We call that infection. But there are also rare mutations in the gene that lead to misfolding. And when that happens, the conformational form, the altered form, accumulates in the CNS, and that leads to your symptoms. So we have 3 types of TSEs. We have infectious or transmissible, right? You acquire some PRPSC from some source. You could eat contaminated beef. You could get a corneal transplant from someone who has a TSC and you don't know it. That has happened many times. And in the old days, we used to purify hormones from people, and that gave other people TSCs. So you basically are getting some PRPSC, and it changes all of your Cs into SC. Then we have familial, where you inherit mutations from your parents in the PRNP gene. And these predispose the protein to misfold. You don't need to acquire any PRPSC. You just make it yourself. And then there's sporadic, where you have a normal gene. You don't get any PRPSC exogenously, and the protein just misfolds. And all of these can be so once you have a PRPSC, then it can be transmitted to animals by ingestion or inoculation. So in humans, we have all three forms: infectious, genetic, or sporadic. And the infectious forms include Kuru, which I'll talk about in a moment. We used to spread it to people by transplantation of infected corneas, hormones, transfusions from patients with CJD before we knew about it. Feeding infected animals to cattle gave them BSE, that's infectious. And variant CJ was people eating BSE beef, contaminated beef. So what's Kuru? Kuru used to be a fatal encephalopathy. It was a TSE in the 4 people of New Guinea. Here's New Guinea here. 30-year incubation period. Carlton Gajdusek, a virologist in the U.S., went to study it, and he found that it was mainly spread among women and children who used to eat the brains of deceased relatives. It was a ritual. After a relative died, the women and children would eat the brain, and that gave them, they probably died of Kuru, and it transmitted to the women and children only. And Gajdusek said, you shouldn't do this. Don't eat the brains, and they stopped, and the disease went away. The first human one discovered. Sporadic, so that was an infectious one because you're eating brain, right? There's sporadic CJ, where the protein just misfolds. It affects 1 to 2 per million humans worldwide, a lot. Between 50 and 70 years of age, this accounts for 65% of human TSEs. It appears without any warning, no epidemiological indications. Patients have normal PRNP genes. And, of course, you can transmit it to others. If you donated a cornea before you knew you had CJ, if you died and donated organs, you could transmit it to other people. And we think Kuru may have been established in New Guinea by eating the brain of that very first person who died of sporadic CJ. So that's sporadic. And then we have familial, where there are mutations in the PRNP gene that cause the protein to misfold. I mean, it doesn't happen right away. It takes between when you're 50 to 70 years of age, but eventually those are causing the misfolding. And these, again, if you develop a sporadic TSC, your organs, your corneas, your blood products can be infectious. You can transmit it to other people. So in the end, if you have a misfolded prion, PRPSC, it can be transmitted to others. And so here are CJD deaths in the U.S. from 79 to 20. So the total deaths here are in blue. This is deaths per million people, so 2.5. And the number of deaths here is 500 or so since 1979. And this is the age-adjusted death rate right there. So it's relatively rare, but this is the U.S. and globally there are many more deaths as well. Now, bovine spongiform encephalopathy, or mad cow disease, was a form of cannibalism, just like Kuru was in people. We fed British cattle, ground up animals, including sheep who had presumably died of scrapie or died before they showed signs of scrapie, so that we introduced the scrapie prions into cows, and they got a form of a TSE called BSE. And this resulted from feeding cows ground up animals. In the 1970s, the methods of preparing this protein supplement changed. It's called meat and bone meal. And this change in the preparation allowed scrapie proteins to survive and pass into cows. So we detected this outbreak. We found cows dying of TSCs, and so we stopped this animal byproduct feeding, and that stopped the outbreak. And there's very good evidence that consumption of BSE infected beef transmitted bovine TSC to humans, and that gave us a new disease called variant CJD, slightly different from CJ. So here are the outbreaks in cattle and humans. So we have cases in cattle here on the left y-axis. This is thousands of cattle with bovine spongiform encephalopathy. You can see peaking around 92. And then it goes down as we change the feed producing process. And then overlapping it, an increase in humans of cases of variant Creutzfeldt-Jakob. And when we figured out what was going on, those went down as well. And then there were a few cases where people who got variant CJ from eating beef, BSE beef, they transmitted it to others by blood products. They're giving blood. And so that's the green ones here. It was estimated that about 1 to 2 million cattle were infected with prions. And the problem is that the incubation time in a cow is about 5 years, and they're usually slaughtered earlier for meat, 2 to 3 years before you can tell that they're sick. So we still have cases of BSE in cattle. These are probably sporadic. The cows just sporadically develop a TSC from misfolded protein. And this is a problem. But we in the U.S. and Canada, we check less than 2% of slaughtered cattle for prions. So there's a chance that this could get back into the food supply. In Japan, they check every head of cattle for prions, but we don't do it here. We now have diagnostic tests, and we can check the cows for that. But you see BSE cases in North America, they're not a lot, right? The blue is Canada, so that's 6 cases in 2006, and the red is the U.S. So 26 cases from 93 to 2018. It only takes one of these to get in the food supply, and it causes a big problem. I want to mention just 2 papers which I blogged about at Virology blog, and this was a case here in 2015 where a man living, I forgot where, somewhere in the Middle East, he developed a TSC and died. And it turned out he had lived in the UK during the BSE outbreak, and he had eaten beef. So that's where he acquired it many years later. And then this one is an incident where an individual walked into an emergency room. He was behaving oddly. He pulled out a gun and shot himself in the head. And all his brains went all over the ER, and turned out he had a prion disease, and so they had to discard all the instruments in the ER because you can't decontaminate. High pH, low pH, temperature, boiling, formaldehyde, none of that will inactivate the infectivity. It's just amazing, just amazing. Anyway, I talked with Richard Knight, was in Manchester a couple of years ago for a meeting, and he knows all about prion diseases. You should check it out, 950. Very interesting guy. So there have been 3 epidemics of Creutzfeldt-Jakob in humans. And again, this is years on the X and number of infected patients on the Y. So the top one is an outbreak by injecting growth hormone into people that had been purified from pituitary glands of people. So we had an outbreak until we realized what was happening. Dura mater grafts, these are a kind of tissue graft, again from people with Creutzfeldt-Jakob causing an outbreak. And then the outbreak from eating BSE beef here. So 3 different epidemics of Creutzfeldt-Jakob. All right, so just for the last few minutes here, I want to talk about the prion species barrier, because this will lead us into chronic wasting disease of cervids. If you inoculate diseased brain material into the same species, you get disease. So if you have a mouse with a TSC and you grind up the brain and put it in another mouse, it will get disease. But inoculation into a different species is either inefficient or non-existent. So basically, we have learned that the prion protein in the inoculum and in the host need to be the same for the misfolded prion to cause misfolding of the cellular protein. So the sequence of the protein is controlling the host range. And here's an example of that. So if you take a hamster with TSE, you purify the PRP-SC and put it in a mouse, no disease. Different species, different sequence of the protein. But if you take a mouse transgenic for the hamster PRNP gene, in other words, you've put the gene into the mouse from hamsters. Now, when you inject the hamster PRPSC, you get disease. Now, bovine PRPSC breaks the rule because it has a broad host range. If you inject it into mice, they get a TSC. And in fact, the bovine PRPSC infects many meat-eating mammals, including humans. And you can transmit it to many mammals. If you feed beef to zoo animals, they're going to get infected beef. They're going to get TSCs. And so some prions can overcome this host range restriction. And this is why BSC is a concern, because the bovine PRPSC has a broad host range. Now, finally, chronic wasting disease is a recently discovered TSC of deer, elk, and moose. It's been found in 27 U.S. states, 4 Canadian provinces, both in free-ranging animals and in commercial captive facilities. In some herds, up to 90% of deer and 60% of elk are positive. And this is a typical animal wasting away and with motor disturbances and so forth. And in wild servants, maybe as high as 15%. And there are many reasons why this is a concern. So this was just discovered in the mid-60s. And of course, the number of animals, these are total and captive and free-ranging, is going up as we discover them. You can see here in the U.S. where U.S. states they have been identified. Why are we worried? Well, first of all, it turns out that grass plants can bind and take up and transport infectious prions. So if you take a plant and you add prions to the soil, the plant will take up the prions, and then if you feed the leaves to an animal, they get infected. So the prions retain infectivity. So we're concerned that the deer prions can be taken up into plants, and then other animals, including humans, if it's a farm, could eat them and get the disease. So that's one thing we're concerned about. However, if you take cervid PRPSC and inject it into a mouse, you don't get disease. So there's a host barrier. If you take a mouse transgenic for cervid PRNP and you inject cervid PRPSC, you get disease. Okay, host range restriction. So what if you make a mouse transgenic for the human PRNP gene? If you inject them with PRPSC from cervix, you don't get any disease. So it suggests that so far the cervid prions are not going to infect us. But the problem is that these deer prions are now known to they can infect other animals. If you put them intracerebrally into cows or raccoons or, I don't know what this is here, moles, meadow voles. It's a meadow vole. They can get infected. So the host range extends beyond deer. So, these animals, if they're grazing in a contaminated pasture, they could pick it up. And maybe it would change as it moves through animals. So far, none of these animals have been shown to be infected in pastures where, cows are grazing. They don't seem to be infected so far. But, all of this can change. So we have to be vigilant, and we have to keep looking for this. And so if you hunt deer, many people do hunt deer. There's this website where they tell you what not to do. Don't consume any animal that acts weird. Right? Wear gloves when you field dress your animal. Bone out the meat. Don't saw through the bone. Don't cut through the brain or spinal cord where there are a lot of prions. Minimize handling of those. Wash up afterwards. Don't eat the brain, spinal cord, eyes, spleen, tonsils, or lymph nodes where there are a lot of prions. And of course, if your animal tests positive after all this, you shouldn't eat it because cooking will not inactivate the infectivity. So it's a little scary, but like anything else, it's manageable. And so not even the vegans are safe. Maybe they get into the plants. So that's my little spiel on prions. I really find them interesting. Let's turn off my black magic here. I really find them interesting. Am I here? No, my picture is not here. Why not? My camera. How do I get my camera back, Vinny? You're seeing my desktop because my camera's on. How do I feed it back into this darn thing here? Black magic. No, that's not it. All right, just give me a second while I figure out the tech here. Camera, Black Magic Design. So camera, switcher window. Why the hell isn't it showing? I never had this happen before after a thingy-mabobber. It looks like I have something here. There I am. I was hiding behind a desktop. I don't know why. Now you should see me. There we go. Can always solve problems, right? Okay, let's get to your questions. Let me look at the bottom here. Maybe you have some prion questions that we can. How are working prion tests? So there's an assay which is kind of a PCR for proteins. So the idea is you take you take a sample from an animal that you think has PRPSC in it, the misfolded protein, and then you add it to normal protein, and you shake them together, and they will. If there's PRPSC in the sample, it will convert the PRP into PRPSC, and you do that multiple cycles. It's called shaking and quaking. You keep adding more, and you go through these cycles of shaking, and then finally you do the proteinase assay to see if you've got PRPSC there. It's a pretty good assay for testing meat. Bad cat. I have deer and moose burger in my freezer, gifted to me, but I'm going to take my chances. Well, if it's prepared from a healthy animal, it shouldn't be a problem. Maybe you could find the provenance of it. It's pretty rare. You could take your chances. It depends. It depends how much you want to eat them. Yeah, I try not to eat much meat anyway, but so that's the prion. Doreen says, "Yikes! Can't wait to relay what I've learned to my zombie fan daughter. No brain eating." How do you destroy the sample when finished? Actually, now some conditions that they've identified that can destroy infectivity, but they're very extreme, and I don't remember them offhand. So, but it can be done. Chris would like to know if you can get prion disease from animal excrement. Well, it is excreted in deer. It is found in deer poop, but you have to ingest it. So, I don't know why you'd be eating poop. That's all I have to say. The prions shed in feces and transmitted through the fecal oral route. Yeah, so, that's not been shown in people because we are not coprophagous, but it certainly is possible in animals. I don't know any documentation of it, though. It's very all right. So MK Painter is saying alkaline hydrolysis. So heating in sodium hydroxide will get rid of it. Mandrake won't swim in fresh water because of Naegleria, the amoeba that can go into your brain. Yep. What else here? No evidence that sheep scrapie can affect humans. No, but remember, the sheep were fed to cows, and the cows got scrapie from the sheep, and then the cow prion, right? Because then what's misfolded in the cow is a cow prion. It's no longer a scrapie prion. And then you eat the cow and you can get it. So indirectly, I guess. Okay. Let's go to the top and start going through, and we'll get back to some of these questions here. All right. And everyone's talking about the RFK Jr. story. So he supposedly had a worm in his brain. He had maybe neurocysticercosis, right? Which we've talked about on TWIP, which happens to people. And you have, but I don't think it makes you become an anti-vaxxer. This is the first time And I don't think you should blame it. He says he's blaming it on everything, blaming everything on it. But it happens in people, and usually they're dead, right? And most of the time they're just nothing is done about it. But we can have Daniel talk about that. But yes, that's what he had, neurocysticercosis. Let's go. All right, I'm getting past all the intros here to pick up. Okay, here's Kang. Could a viral infection induce enough interferon to prevent the second infection? Ah, that was from last week, Kang. Remember, you never got it answered. So yes, for sure, if you have a virus that's good at interfering, good at inducing interferon, and there are some, and then if you happen to be co-infected, that could inhibit the other virus. Now, a practical illustration of that was the beginning of COVID. Kostya Chumikov said, it's possible that getting OPV will prevent COVID, because when he was a child, his mother gave him many doses of OPV. It's oral, right? Every year to prevent him from getting influenza and other respiratory diseases. And she published papers on this. She was a virologist, and she said it worked. And so probably the effect is inducing interferon, which then inhibits the other viruses. So it's a transient effect, right? Maybe, I don't know how long it would last, and you would have to take multiple doses throughout the year. And Kostya was trying to do a clinical trial, but never could get it off the ground, never could get the funds to do that. But yeah, you could be co-infected, and one is a good interferon inducer. And then, yes, there are some viruses that are resistant. They don't either are resistant or they don't induce it very well because they encode gene products that are antagonists of interferon. And so that would allow the other virus to reproduce better, right? So you have two scenarios. If you have a good inducer of interferon and another virus comes in that's sensitive to interferon, that virus is going to be inhibited. But if the first virus inhibits an interferon response, then the second virus gets a free ride and can go crazy. Yep. Crazy stuff. Here's a link to the worm ate part of my brain. I don't think it ate part of his brain. I mean, there's a hole where the worm is, yeah, but I don't think the worm ate it. But you could imagine what you want. Sure. Yes, I saw this. Physics professor Sabine Hassenfelder did a short video on H5N1. Why is a physics professor doing this? I know Sabine Hassenfelder's work, okay? Wasn't too terrible. That's great endorsement. It wasn't too terrible. Well, do you want to hear Richard Webby, who's worked on flu his whole life, or do you want to hear a physicist? I'd rather hear Richard Webby. So listen to TWIV, episode 1113, releasing on Sunday, May 12th. Rach from Isle of Man. AstraZeneca has discontinued a lot of anti-vax comments about it killing loads of people in media comments, ATM. I had AstraZeneca and I'm okay. It didn't kill loads of people. It's just wrong. The vaccine itself, this is a common misconception that people spew, that vaccines are killing loads of people. No, they don't. First of all, they were clinically tried in tens of thousands of people, and then they were subsequently given to millions of people. And people are not dying of them. We would know that. There are some rare side effects, right? So the AstraZeneca adenovirus vectored vaccine caused blood clots in a number of people, which we didn't see till we got it in 100,000 people because it was rare. And some of them died. Yes. And so in the U.S., we discontinued that. But it's not killing loads of people for sure. Absolutely not. There's no basis for that. what viral diseases can one catch from urine? The cytomegalovirus is one that comes to mind. And other viruses are shed in urine. Some of the polyomaviruses like BK and JC are shed in urine. The extent to which that causes transmission isn't clear because it's probably respiratory transmitted also. So I say the big one is CMV. HIV is not transmitted by urine. Let me just check. There are lots of viruses in urine, of course, but not clear that, like human papillomaviruses can be found. Low risk. So that could certainly be, because that could be sexually transmitted, right? But I think cytomegalovirus would be my top choice. Krista says, does the transmission of the recent bird flu to other species follow that of SARS-CoV-2? I don't know what you mean by follow that. I'm not aware that. So deer and mink were big, were infected quite often with SARS-CoV-2. I should say very effectively. And I'm not aware that H5N1 is in deer. Could be wrong. But I wouldn't say that it follows. There are a lot of mammals out there that are in contact with birds, and they're going to be infected. And the animals got it from humans. So I think it's going to be a different subset, right? Pete says, last Thursday, Eddie Holmes was the Royal Society Croonian Prize Lecturer. A tour de force hour on a journey through the virosphere would pair with Vincent's first virology course lecture. It's a good suggestion. Sounds great. Now we have all the where I am from. Carol says we had a raccoon partying in our attic, which the termite company was able to trap. Cleanup by the termite company was extensive with lots of poop and pee. What viruses should we worry about? just rabies. I should test it to see if it's rabies positive. I mean, I think the only concern would be if the raccoon bit or scratched someone, then it's likely to transmit rabies. But it should be tested for sure. Hello, MK Painter. Thank you for your support of science communication. I don't know what that word is. I can't copy it and paste it into the translator. Is it Cyrillic? Oh, no. Maybe you could translate it. Thank you for your support, though. Here's the link to, I suppose, the Eddie Holmes lecture. And Tom notes that Eddie was on TWIV twice, 940 and 1019. So 940 we talked to him while he was in Australia. Then 1019, I had him at ASM Microbe in person. Prions are also birds. A small petrel of southern seas having a wide bill fringed with comb-like plates for feeding on planktonic. Oh, a prion is a bird. Interesting. So I wonder if Stanley knew that. Because if you look it up, it all comes up, the misfolded protein. Let's put prion bird in. How'd you find that, Peak? Yeah, there it is. It's a bird. Prions are whale birds, or small petrels in blah, blah, blah. Small petrels. Very cool. All right, now we know where the prion road came from. Did you ever get a COVID booster, or are you still at 3 shots? Well, remember the third was called the booster. I got a fourth one. I got the new Omicron last last fall. But that's all I'm going to do. Visto says, how specific are prions? Can they only corrupt the exact same protein? Yeah, most of them can only corrupt the exact same protein, but there's some exceptions, right? Because the bovine can apparently corrupt different prion proteins like humans and other mammals, right? So there are always exceptions, yeah. I don't know if we said hello to Peter from Boulder, Colorado. Hello. And Elmug is from Pueblo, Colorado. Patricia writes, new fear unlocked. Yeah, sorry, Patricia. I used to have prions, but they're not pathogenic. They are different states of a protein that have different functions. All right, so in one state, a protein has one function, and then in a different state, it has a different function. It's not pathogenic. Prions are pathogenic in mammals. Well, ostriches can get TSEs. Let's look it up. Ostrich TSE. I get lots of cashmere. Can an ostrich get prions? What animals can get prion diseases? Oh, that doesn't help. It's all the ones I told you. It's all, what diseases do ostriches carry? All different viral diseases. Diseases associated with ostrich products. Let's see if prion. Well, it's one of the keywords in this article. There's little evidence to suggest that ostriches could play a role in the epidemiology of TSEs. Well, maybe they get them. Anyway, it's possible. You know, you can, whenever you say it's only in something, you're going to get in trouble because there are always exceptions, right? How are prions taken up by the cell? That's a good question. Not clear that it has to be taken up because the prion is on the surface, right? It's a surface protein, so it can just misfold on the surface. The question is, if you ingest the prion, right? If you eat BSE contaminated hamburger, how does it get from your stomach to your central nervous system? And it's not clear how that happens. Yeah, nobody really understands that. As a matter of interest, prion is a last name. Just looked up some phone listings online, probably where the street name comes from. Okay, mystery solved. By the way, if you lived in the UK in the 90s when we had this BSE outbreak, you cannot give blood, ever. Can't give blood. Now, someone is going to tell me I did live and I gave blood. Okay, but officially, you're not supposed to give blood. Pete says in the 90s in the UK, I found the journalists and politicians couldn't communicate the uncertainties of science as the BSE crisis played. Probably they didn't know it. But listen to Richard Knight. He's very good. You can find some other talks by him online. It's really good. Intersoul said, I used prion in that word game. It wasn't the best first guess. The answer was vaguely relevant to prions, though. Mark Martin, time to plate, be back. And by the way, I was in grad school when Stan Prusner came to talk in the early days of his work. Charlie Yanofsky was a big booster of him. Yeah. He was a hero. He was a hero. He had this idea, and everyone said he was full of it. They said, Stan, you're wrong. It's a virus. I mean, they insulted him. You should read his biography. He talks about it. And he just stuck to his guns and got the evidence that proved it. And now it's accepted that it's a thing. These are infectious proteins. And yeah, actually the virologists came to his rescue. They supported his idea. Many of them realized it wasn't a virus, but it was an infectious etiology. And that's why it's in my virology course, because the virologists supported it. And I always like that too. Pete remembers his biology teacher mentioning the mystery of Scrapie in 1970. Wasim said, a perfect topic for me, trained as a veterinary virologist and working as a field vet in northern Himalayan mountains. Wow. that where you're off? That's where you are now? It's pretty far away. And yes, there was certainly initial skepticism to his proposal. People say it's contaminated with viral DNA. It's contamination. But the genetic experiment, you knock out the prion protein gene, you don't get TSE when you inoculate. That's it. That really does it. Professor, may I ask, what is the difference between positive and negative sense RNA viruses? Lecture number 3. A positive sense RNA virus has RNA, which is the same polarity as messenger RNA, which means it can be translated into protein. And the minus strand is the opposite sense. So negative strand viruses, their genome cannot be translated. It's the complement of mRNA. Plus strand RNA viruses have a plus strand genome, which in theory can be translated. Most of them are upon entry of the plus RNA into the cell, but not every one. Like retrovirus genome is plus RNA, but it is not translated when it gets into the cell. It is reverse transcribed into DNA. So that's what a plus and minus strand RNA virus is. We had a surgical patient that created a challenge because he had a prion disease and he required brain surgery. Decontaminating the equipment post-surgery was also challenging back then. Yes. Well, in that emergency room paper, which was a number of years ago, they had to throw out the equipment. They didn't have yet methods developed for it. David says tardigrades use intrinsically disordered proteins to survive desiccation. Well, I didn't know that. And maybe Mark can comment on that. Mark seems to be an expert on tardigrades. Very Nika says, I guess there is a positive side to prions. Maybe they can survive in outer space long enough to transform life. Sorry, I've been watching many space videos lately. I don't know how we could transform life. Is CTE disease of footballers prion related? Chronic traumatic encephalopathy, right. I don't believe it's prion related. But so Prusner says all of the chronic neurological degenerative diseases, Alzheimer's, Parkinson's, others that I can't remember, he says they're all prion disease. They all involve misfolding and deposition of plaques in the brain. Not everyone agrees with him, including Richard Knight, who I interviewed there. But it's an idea that protein misfolding is the basis of many diseases. And if not a prion, maybe some other protein. Could a GMO protease digest the misfolded protein? Well, yes, but you don't want to give people proteases, right? Maybe you're thinking of environmental decontamination. No, remember, it's not completely digested. Although that's a good question. I don't know, is the digested form pathogenic or not? I'm sure that experiment has been done. If you digest PRPSC with proteinase K and then inject it into an animal, will they get prion disease? I don't know the answer to that. I'd guess not, because you're taking away a third of the protein, but I just don't know. Can you treat a prion infection? No. No treatment. No, we have no drugs that work. And by the way, this is your protein, so you don't make antibodies. You don't make an immune response. You don't make T cells, because it's you itself. And there's no treatment. We have no drugs that will inhibit their production. Although you would think, in theory, you could make molecules that bind PRPC and prevent it from misfolding, right? But I think it's really hard to get those. Right, but that's what people are doing. Yeah. Laurie says, I remember the ban on fetal bovine serum for tissue culture from France and the UK in the 80s and 90s because of BSE. Right. We don't want to ship that stuff everywhere else. What is it that Richard Knight told me? Very interesting. So it's quite a market for deer urine. Did you know? because hunters, they buy it and they sprinkle it to attract deer, and then they shoot them, of course. And then that deer urine could have prions in it, so it's a potential source of infection, right? So that they're worried about deer urine. Simon says, we had massive culling of cattle in the UK during the BSE outbreak. It was quite horrible. Yeah. Many millions of cattle were culled. It's like millions of pigs were culled in Malaysia during the Nipah outbreak. Really, really horrible, yes. Who else remembers in 1990 John Gummer, UK agriculture minister, stuffing a burger into his young daughter in front of the TV cameras? Beef is safe. I don't, but oh my gosh. Oh my gosh. Yes, I could see them doing that because they wanted to assure people that the beef is safe, when in fact it might not have been. It was quite an era. A lot of things happening and lots of people's reputations ruined because they didn't know what they were talking about. No, does it sound familiar? What were they doing feeding meat to grass-eating animals? Well, if you feed meat to cows, it grows faster, and you could get it to market quicker, and you save money, right? That's what it's all about. But yes, they're grass-eating animals. They feed them meat, and they grow really fast. There you go. Running a business. Yep. That's it. Amino acids. Amino acids. Can you make an artificial nucleon? I don't know what you mean by that. You mean synthesize the protein or make a new sequence? I don't know that you'd know what to make, right? You could certainly make the protein, but here's the thing. It's the PRPC protein sequence, amino acid sequence. It's the same sequence, folded properly and misfolded. It's the same amino acid sequence. So what causes it to misfold? Especially if there are no amino acid sequence differences, as I've just said in most cases. Don't know what would happen. It just has an inherent intrinsic ability to misfold in a specific way. Proteins do that. They can exist in multiple conformations. And this one just happens to be bad news when it misfolds. Yeah. I think so. Yeah. Mandrake says, I think sodium hydroxide. How to inactivate prions. Use an autoclave under severe conditions, 134 C, 18 minutes, sodium hydroxide, 1 normal, SDS, 30%, and bleach. Oh my God, 20,000 parts per million. That's really extreme. So you need to use an autoclave. So large equipment is very hard. You can't get it in an autoclave. And if you could, you'd break it anyway. Is a vaccine possible, at least theoretically, against prions? I suppose if you could if you can induce antibodies to the conformationally altered protein, right, which should be possible. There should be different epitopes there. But I don't know. I've never read anybody doing it. It's not an option, given the rare incidence rates. The situation might be different for familial prion diseases, which can be diagnosed long before clinical symptoms start. So there's actually an article here in Cell and Tissue Research 2023. Vaccines for prion diseases, a realistic goal? ? There's currently a strong push to develop vaccines against chronic wasting disease to be used in wildlife. But the immune response does not develop a bona fide response against prion infection. And because the proteins share identical protein primary structures, prions seem not to represent a trigger for immune responses. But there are some other therapies being addressed. So let me put this in the show notes here. There you go. There's a link to the article. Good question, very Nika. Very good. So it's not easy, but people are working on it because there's no solution. And the wildlife thing is a problem, right? If you could feed bait to deer with a vaccine, that would be cool. Can accidentally occur a new prion in the lab experiments. I don't know how you could do that, right? All right, so I can see where if you're doing an experiment where you're taking a protein and you're changing every amino acid, right? Maybe you would end up with a sequence that could misfold, but you're not injecting it into animals, so it's you're never going to know. So I would say theoretically it's possible, but nobody really is trying to do that. Why do prions occur in evolution? Okay, so in yeast, there are prions that have two states, two conformational states, just like this. And both states have a function. And so the idea is when the yeast are stressed, the prion flips conformation and it has a new function where it can deal with the source of the stress, right? So that's the idea. But somehow they have acquired this pathogenic property. They don't seem to be essential. You can delete the gene in mice and the mice are fine. So it could be a remnant of an older function, right? And of course, yeast are old, right? Could be a relic of an RNA world. Well, no, it's a protein. So it's not a relic of an RNA world. It's a protein world, but it may also still be very old, and it may have had a good role, like in yeast, where it serves two functions. And then the protein was conserved for whatever reason, and then it turns out that when it flips in a mammal, it causes problems. So Lisa is here with Christopher. Hello, Lisa. Nice to meet you. It's alarming. Yeah. The other day, a beautiful hawk crashed into our house and died. What do you think? H5N1. I mean, birds hit windows all the time, right? A hawk shouldn't hit a building. Yeah. It's possible. I don't know. You have to check. Do prions break cameras? Not that I am aware of. Break. I don't know what actually you mean by that. Break cameras. Maybe it's a joke that I don't understand. Chris would like to know if you can get prion disease from animal. No, you don't. I'm not aware of it. As I've said, that you can get it from excrement. Yeah, you have to eat it, right? Humans don't do that, as far as I know. My dog would eat poop if we'd let him, and he could transmit it to us. Dogs can eat deer poop. They do at the local dog park. Yeah, I know that for a fact that dogs would like to eat any poop. So I don't know if dogs can get chronic wasting disease. Can dogs get? It's a good question. There's no evidence that dogs can be infected. However, it is best to avoid feeding brain and spinal cord tissues from killed game to dogs. So prions, CWD prions can be excreted in saliva, urine, and manure of infected animals. So theoretically possible. It is a easy experiment to do. Well, it's not so easy. To try and infect a dog in the laboratory. I don't want to do that because I don't want to infect dogs, but you could ask, or so here's what you could do. You could take the dog prion gene and put it in a mouse, make a transgenic mouse, and then infect the mouse with chronic wasting disease prions and see if it gets infected. That would be a good experiment to do without harming any dogs. Yeah. Good questions, folks. Yes, Laurie says, because I lived in the UK in the early 90s, I am unable to donate blood because of possible prion exposures. That's right. Any angry penguin, any known prions in dogs or cats? In cats, for sure, there are feline transmissible encephalopathies. Yeah, but as we just said, I'm not aware of any in dogs. Can deer transmit disease to dogs? No, that's a different disease. So no cases of dogs becoming infected. And some studies shown that they seem to not be infected. They call it immune here, but I would say that it's probably the species barrier. Yeah. Wild mushrooms might pick up prions from cow pats and deer pellets. Yep. Don't pick wild mushrooms. Pasteurize your milk. It's a tough world. Yeah, I know. And you want to, live wild, but damn, it's a little conspiring. We have 210 listeners. Thank you so much for coming. 166 likes, which is not bad, but we'd like you to hit the like button, get it close to 200 so more people will hear about this program. Because you learn cool things, right? I know what floats your boat. I mean, prions for sure. And the last mini lecture, which may or may not be next week, I don't know, is going to be about therapeutic viruses, using viruses to treat disease. That's fun too. Doreen, thank you for your contribution to science communication. My mistake to hear this and TWIP on hookworm on the same day. I've exceeded my daily gross out tolerance. Yeah, the kid with a leech in his throat. Oh my gosh. And the worms ate into his brain. Is that a Pink Floyd line? Really? And the worms ate. Yeah, it comes right up. Hey, you. Hey, you. I'm not a good. Hey, you out there in the cold, getting lonely, getting old. And where's the worm thing here? Let's find it. Here. The wall was too high, as you can see. No matter how he tried, he could not break free, and the worms ate into his brain. Pink Floyd is great, right? I mean, I love all kinds of music, modern music, but Pink Floyd is great. Love it. Wasim, veterinary virologist. Okay, so Wasim knows what they're talking about. We need to have a strong differential diagnosis from animal rabies, as we still need a brain sample for testing. Lately, muzzle samples from cattle have also been used for diagnosis of rabies. Veterinary virologist. Talking about Carol's raccoon in the attic. Luis, back to viruses and H5N1. Are there avian H5N1 antigens present that may help induce seroconversion of the animal that drinks pasteurized milk? There should not be antigens. All right, so if there are virus particles in milk, and some people have said there are no infectious particles, but I have to ask Webby about that on Friday. There probably is not enough to induce an antigenic response, right? That we actually talked about that on Clinical Update a few weeks ago. So I don't think it would seroconvert, no. Could prion originate from alkaline molds in mite guts feeding on linoleum? I don't think so. No. It's a normal protein that's misfolding, right? Sheep fed to cows. It's terrible that cows are fed what they don't normally eat, against nature. So, yeah, that's business, as Lori said. It's business. Last Light says, how does one misfolded protein provoke other proteins to misfold? How does it perpetuate misfolding? Well, it's a great question. It is a great question, and we don't know how that happens. We have structures of both, but we do not know how one causes the conversion of the other. And Luis says, great question. Is there a different energy between the two forms? If so, where does the energy come from? So these are good questions. No answers. Yep. All right, back to hold on a second. Okay. A different energy between the two forms. Okay, back to RFK. There are a few species of brainworms. Did RFK say if it's tapeworm or Baleascaris? I don't know. Don't know the answer to that. Let's look it up. Pork tapeworm. He had pork tapeworm. Neurocysticercosis. Yep, pork tapeworm. Oh my God. Read that the worm would just die in the brain area and a cyst would form. That's correct. That's correct. Vincent, the AZ vaccine was discontinued because it had not been updated to new variants in countries focused on mRNA vaccines. But there was also in the U.S. Not used because of the clotting issue. It was discontinued here before variant vaccines were needed. COVID vaccines were probably the most tested medicine in history. It was tested a lot. I would think that most virologists are extra careful when using public washrooms. I don't know. I think most virologists are humans and they do human things like everybody else. Kang says, what's the origin of prions? Chance misfolding. It could be that, yes, because it's the same protein, it's the same amino acid sequence, right? So it could be that once, many years ago, it was a chance misfolding and it had a biological benefit. And it could have happened in an ancient organism like yeast, and then it was just maintained. So that's what, yeah, I think chance misfolding that led to a biological benefit, right? Hat Frinson from Clinical Update. Nasal neomycin. Neomycin is a topical sensitizer that we do see in practice. We frequently ask patients to avoid and use topical antibiotics without neomycin. Yeah, so seems to induce interferon, right? There was a paper in PNAS, it's a broad antiviral because of that. But Daniel is like, don't want people putting it in their noses. Okay, so Veronika said the word that I asked about before is polite hello in Russian. Okay, thank you very much. And the link was for the virology class lecture on prions. Okay. Thank you. I didn't know. I thought it would be Eddie Holmes. Sorry about that. Let me see if I can. So Rima says shaking and quaking. Okay. Let me tell you. Let me see if I have a slide about shaking and quaking, right? That would be lecture 24. I didn't show it this year, but I may have just skipped it. Let's see. Because I skipped. Here it is. I skipped it. Unskip slide. And let's take a look at this. Okay. We have to go back to sharing the screen. And so here's the assay, detection of prions in nasal brushings and urine. So you can brush the olfactory neuroepithelium. You could do the same in cows. You could take urine. So basically, you take a sample. Well, where are we going to start here? So in this assay, what you've done is you've taken PRP-SC, which are oligomeric. So these are the misfolded proteins. They're forming these long aggregates. And then the PRP-C are monomers. And if you incubate them with shaking, okay, on a shaker, then these PRP-Cs become, I'm not showing it to you. I'm sorry. Damn. Let me start over. I'm waiting for it to come up. So this is the assay. You take misfolded prions, which form these aggregates, and you mix them with monomers of PRP-C. You incubate them with shaking, and then the monomers form oligomers. And then you add more monomers, and they get longer and longer. So if you have an initial sample with PRPSC in it, and you add some monomers, they're going to form longer and longer oligomers with each cycle. You do 96 cycles, and then you add some more PRPC, and then you do another 96 cycles, and then that's how you do the assay. So that's shaking and quaking. I don't know where the quaking part is. It's just a mix. It's a mixture of shaking and other kinds of movements on the incubator, I believe. And so hopefully you saw that, because I didn't share it. That's why. Jesus, I didn't share it. Here, I'm going to go through it again. I presume you can see it now. If you have a sample with PRPSC, which is aggregated, and you add fresh PRPC and you shake it, the PRPC will be misfolded by the PRPSC and form long aggregates. And if you do this for many cycles, you get longer and longer aggregates. And then the quaking, you shake it really hard, and the quaking breaks up the oligomers, and with the addition of new PRPC, they can make new oligomers upon shaking. So that's where the shaking and quaking comes from. And so that's kind of a amplification protocol for protein. Yeah, there's an ultrasound step to break up these long monomers, and then you add more PRPC. And that point is, you bring it up to a level of detection where you can do a Western blot. And so this is now being applied to detect, in cows, in people. That's shaking and quaking. We discussed bird flu. Nobody's asked if I was worried, but Friday, Susan, we're having an expert on TWIV. You can listen to that, right? Do prions suggest that non-genetic information can be inherited through proteins? No, remember, these proteins have a gene encoding them. Right, so that's what's being inherited, not the protein itself. Proteins are just pathogenic. Daniel's had 4 shots. I will get another. Heard that another is available. Yeah, you can get another one. Yep. Yeah, unfortunately, prions can be taken up by plant roots if a prion-positive animal poops or urinates where food is going. Right, that's the picture I showed you. Can prions get prions? Well, if a normal prion encounters a misfolded one, it becomes misfolded. So that's the closest. Okay, Tom found the only occurrence of a TSE in an avian species was reported from a flock of ostriches in a German zoo. Okay, got it. Were subviral agents known before Stanley? No. You mean Stanley? Wait, Stanley Prusiner. Subviral, like helper-dependent viruses, were known, yes, but not protein agents, no. Prion to brain. It's like calling, right? Prion to brain. Maybe shortly after eating, prions move somehow from the mouth area to the brain. Oh, that's a possibility, right? From the mouth. There are innervations that they could move, yeah. Or from the intestine. I think that would be fine. Interesting. Carol wants to know, should I ask the termite company to test the excrement for rabies? Oh, I think if you saw the veterinary person there before from the Himalayas, he said we need brain samples. Well, you could ask them if they tested for rabies. That would be, you don't have to test the excrement. just ask them what they did with the carcass if they tested it. Maybe that's a routine thing that happens when they catch raccoons in houses, right, to see if they have rabies. Oh, I used to eat a tremendous amount when younger and often wondered if I had hookworms since we all went barefoot. Well, it depends. The hookworms come from other humans and you walk in areas where they have defecated, right? So before outhouses, people would get hookworm because people would go behind a tree and defecate, and then other people later would go and walk there barefoot and they'd pick up the hookworms from them. And so they found that if you dig a 6-foot pit and put an outhouse on top, the worms can't crawl 6 feet up. So that's how they defeated the hookworm. So it depends when you lived, if people were still defecating on the ground, right? That's it. Would you explain how p-values are calculated? Nope. I don't want to explain how p-values are calculated because I always use a formula, and I always avoided it because, yeah, it's calculus basically. So you have to get areas under curves, but I don't know exactly how it's done. I'm sorry. I just don't know it. Patricia hates talking about prions. I like it because it's quite an interesting thing. What biosafety level is needed for work on prions? It's BSL-2 because you have to ingest it or inject it to get infected. So it's not going to aerosolize and infect you. But let's look it up. Prion work, what biosafety level? 2 or 3. Most human prions, 3. BSE prions, BSL-2. Okay, so it's 2 or 3. Am I wrong? What's the difference between alpha and beta coronaviruses? Well, it's basically classification, right? The genome differences put them in different genera, right? Let's see if I can get a slide for you. I used to have this in my... let's go to viral zone. That's the best place. We'll look under single-stranded RNA viruses, nidovirales, coronaviridae. So you have, I wonder what the level is there. So the genus, there are 4 genera, alpha, beta, gamma, and delta coronaviruses. And the alphas have some human viruses in them, like the common cold coronaviruses. There's some feline coronaviruses there, and then the beta coronaviruses are, SARS-CoV-2, other human common cold coronaviruses, and MERS-CoV, but they're differentiated based on genome sequence, and they end up having different properties. There's still people who believe in SARS-CoV-2 was engineered. How should I persuade them? Well, there's no evidence for it being engineered. There is good evidence that it arose from nature. We have lots of lines of evidence. You can have them listen to Michael Warby TWIV or Eddie Holmes TWIV. But that's the thing, for example, the FBI and the DOE said it could have been a lab leak, but they have low to moderate confidence. And they, in their reports, if you read them, they say there is no evidence that the lab in Wuhan was working with any virus closely enough related to SARS-CoV-2 to have been modified and released. So I don't know what they're basing, weak to moderate evidence on, because there's plenty of evidence that it came from nature. And it's hard evidence to explain to people, but no human pandemic or epidemic virus came from a lab. They always come from nature. Every virus that you can think of that infects humans came from a non-human animal. And so this is the same. The early epicenter was the seafood market in Wuhan, not the Institute of Virology, which is what you would expect if, in fact, the infections came from there. It would be an epicenter of many cases around it early on, but they're not there. They're across the river at the seafood market, which is where environmental samples have shown the virus to be present. So good evidence for natural origin. And today, if you look on Twitter, Richard Ebright made nasty comments about me today. He called me all kinds of names on Twitter because I'm lying to people about the origin of SARS-CoV-2. I'm not lying. I'm just following the science, folks. There's no reason to lie about that. It's crazy. But, when you're backed in a corner and you don't have anything else, you say that people are lying, they're fraud, all kinds of ad hominem stuff that is not science. When you heard of prion disease in the 80s and 90s, did you become vegetarian? No, I didn't eat meat for a brief period. I did eat fish for 2 years or so, mainly because I didn't like how the animals were treated. That's the main. I'm not afraid of prions because it's such a rare event. I don't, you have to make risk benefit choices in life, and I am I'm not going to be afraid of prions. No. Let's see here. Lisa says there's a great New Yorker article about BSE by John Lanchester called a new kind of contagion. 96. Very interesting about separation of FDA and USDA type responsibilities. Oh, this is good to know. Thank you for recommending. I'll check it out. Let's look here. A new kind of contagion. Yeah, there it is. New Yorker. I got the PDF right here with a nice picture of a cow and the brain that I showed you with the holes in it. You see, I tell you the right stuff, folks. Pete says in the UK 90s, I did reduce beef consumption right down, but only chicken and lamb until risk lower in the 2000s. Well, it was much higher back then because, many cows entered the food chain. And so then they eliminated that source. So now there's no risk. But remember, cows can get sporadic BSE, and they could, in theory, enter the food chain. No solution for prions. Mother Nature is a cruel mistress. Mother Nature is tough. Nature is not human-hearted, for sure. Although I would say humans are not human-hearted. But there are some human-hearted people in the world. But just humanity in general, is it human-hearted? Look how nasty people are to other people. So I think you include humans in nature. West Nile can also make birds hit windows. Yeah, I would say that that's correct. Yeah. Yep. Mark says, if catalytic antibodies can recognize intermediate states, maybe a monoclonal could prevent the switching. Yeah, that could be. The thing is, Mark, you have a lot of PRPC in you. So you need a lot of monoclonal, I think, to block them all. I'm not sure that's because early in a virus infection, you have antibodies that are keeping the viral loads low, but not getting rid of everything. So I don't know how that would work for a cellular protein. Just thinking about it. Yeah, Veronica says birds of prey are super susceptible to West Nile, so that hawk may have had that. Yep. Nature is not human-hearted. Abdul, thank you for your support of science communication and the promulgation of the right stuff. Thank you. We appreciate it. Is it possible to tell if transmission is airborne or too hard to tell with mostly animals having it? Well, there's no evidence for that kind of transmission, right? Which has very specific epidemiological signatures. Whereas if you, you have an outbreak associated with ingestion of beef, it's the eating that's doing that, right? So an airborne component would have a very different pattern. You would have multiple, say, infections in households, for example. We don't see that at all. So when there's one person in a household with Creutzfeldt-Jakob, you don't see other people getting infected either. So that's my thought about that. Well, two listening said the ban has been lifted, but I know someone who lived there in that year and she cannot give blood. But maybe she's not informed. I don't know. Well, let's see. Can I give blood? Blood if I lived in UK in the during mad cow disease. The FDA updated its guidelines, and it means most people have traveled, lived, or had a transfusion in the UK from 1980 to the present are now eligible to donate blood as long as they meet all other donor eligibility requirements. Well, I have to send that to my friend. Let's see. Maybe she knows that. Thank you very much. Appreciate that. Now I am corrected. Have you seen the researcher who raised the leech in his nose? Isn't that what they talked about on TWIP? Hey, you. Oh, boy. Let's see. Microbe TV. Now I have to find TWIP. Let's see. TWIP. This is, all right, it's hard. It's hard to find. All right, just be patient. This Week in Parasitism. The last episode was hookworms and a leech. There's no links to it, so I'd have to listen to it, and I don't know what they said. But well, we could look at the show notes for TWIP. That's very easy to bring up. That's 234. Now let's see what they said, because I wasn't on that one. Hookworm, nothing in the show notes. So maybe it was in TWIP, Mark, because I remember them talking about a leech, and it has to be. Okay, MK Painter says work with prions would depend on risk assessment, most likely BSL-2, possibly with 3 practices. So it's iffy. Don't pick wild mushrooms. Do not say that near Slavic, Finno-Ugric people. We love this stuff. Well, you got to make your own risk assessments, right? You decide. There hasn't been any deer to human transmission so far. So that's good. So you could probably pick your wild mushrooms, but I will just buy mine at the grocery store because they've been grown indoors, right? The Finnish even eat a fungus that is deadly if not cooked properly. Finns are tough people. Yep, prions and cats. Yep, good to know. Louise says there's no infectious virus in pasteurized milk, but raw milk is an infected. Different ballgame. Yeah, unpasteurized milk. That's a question I will ask Dr. Webby Friday. Is unpasteurized milk a problem? Yeah. Very neat. I heard of a story of a whole family getting infected with hantavirus while picking mushrooms in a forest. So yeah, be careful. So they probably got it from rodents, as that would be the carriers of that virus. You have to be careful, yeah. Do we know that prions refold existing proteins, or do they influence new protein synthesis? No, they refold existing proteins. You can mix them in vitro and they will refold existing proteins. Yeah. I'm wondering if AlphaFold prediction program predicts both forms of the prion. I don't know. It's a good question. I don't know anyone who has tried it, but you could look it up to see. That's interesting. If both conformers would be detected. Yeah, predicted. Neat. Maybe this is something we need a treatment for. It's lethal and bad and endemic in some countries. Yeah. there is a vaccine. So that's the solution, to vaccinate, not just people, but wild animals. So, that should be easier than developing a treatment. Yeah. Problem is, once you have symptoms, right, then it's a problem. So if you get bitten by a rabid animal, you need to find out, and then you can start a course of immunizations that will interrupt entry into the central nervous system, and you won't get the signs of rabies. So, the vaccine is really good in that sense. Spamlet says it's incredible that amino acid sequences fold to make enzymes and functioning structures at all. Seems extremely unlikely that both an enzyme and a substrate would ever exist de novo at the same time. Evolution can do a lot of things, man. Holy cow. Evolution has a lot of time and a lot of space to work in. And stuff that seems to us unreasonable, evolution is nothing. It's a day at the beach. Yeah, so Tom explained this shaking and quaking better than I did. Is an amplification protocol, quite clever. There's ultrasound step to fragment the newly converted aggregates, right? Yep. Yep. So the etiology would be sheep wool to mold to mite to alkaline misfolding of wool protein. I don't know what you're talking about. With RFK Jr., the tapeworm was eating his brain. It finally encountered an idea that even a tapeworm couldn't swallow, and it killed it. Very good. Hari. Hello, Hari. Nice to see you. Have cows and milk been tested for other flu before? That is, do we know if H5 is unusual or not, or do we not know? That's a good question. I don't know if they have tested, but in the case of the cows, they were having issues. They were making less milk, and they seemed a little sick, so they tested them and they found it. So I'm not sure that they routinely test them. Yeah. Why does the brainworm die? That's a question for Dixon. Next time we have him as a guest, we ask him that. I'm not sure. Maybe it doesn't get the nutrients it needs up there. It's an accident that it gets in there. Or it's not where it wants to be. It wants to be in the gut, right? Okay. Did the zombies in Night of the Living Dead have misfolded prions? It was a movie. They were just zombies. But they could have, but why all of them at once, right? Did they, it's a long incubation period. It's pretty rare, so it's unlikely that you'd have so many zombies in one town at the same time. Are veggies safe? Yes or no? Yes, veggies are safe if they're prepared properly, right? I mean, if they're washed with bacteria-containing water, no, especially if you don't cook them. But in general, they're safe. There's no evidence that there are prions in plants that we have to worry about. But it's a possibility, right? It's weird that RFK Jr. as an environmentalist so said, but he ate so much tuna sushi he has high mercury levels, though he complains it's in vaccines. Okay. Daniel says, called you names, Latin names. One of these silly things you call someone. Look it up on Twitter. Look for eBright. He just tweeted yesterday or today. Yeah. That's the best explanation I have heard, Vinny. There are some human-hearted people out there. Yeah, there are some. I think all of you are probably human-hearted. But if you look at humanity as a whole, not everyone is human-hearted. I mean, there are people who, for example, want to rule other people and tell them what to do. And there are people who kill other people and do all kinds of nasty things. And, life is amazing. Why would you do that? Figure out your problems. Solve them. Yeah, it's hard. But killing is too easy, right? Virus in filtered milk. Well, the virus is going to pass through the filter. This has to be pasteurized. Prions in cats are maybe a good reason to revisit kissing my cat on the mouth. I don't think it's a good idea. Daniel said it would be fine, but I don't think you're going to get prion disease from kissing your cat. No. Fisto said, did you say earlier that the discoverer of prions later contracted prion disease? No. Stan Prusiner did not. He didn't discover them. He discovered that it was a protein, right? The infectious agent was a protein, but he did not die of prion disease, no. Evolution rocks, shakes, and quakes. It sure does, Luis. That's good. That's really good. The idea of spreading by changing the geometry of something was thought up by a physical chemistry theoretician in England, I think, the 70s. Scary we have found something that can do that. Yeah, people have probably thought of all possibilities, and then you just show it in nature, yeah. I had raw juice last week and got food poisoning, if you consider that safe. No, food poisoning is not good, right? It harms you. There's no reason to be unhealthy, so it's not safe. Ariozone says, do you think Dixon knows how venom from a rattlesnake bite in humans affects blood and tissues? This is assuming that the human receives anti-venom but is suffering significantly. He might. We can just ask him next time he's here. Just remember, write it down. All right, almost wrapped up here, folks. 4 minutes. Human-hearted people would press the like button. Actually, we're pretty good. 199 out of 208. I'm proud of you, folks. Thank you. Yeah, love you all. You can't say I'm human-hearted. You have to have other people say that you're human-hearted, right? Hi, Dr. Racaniello. Just wanted to say I'm a big fan of your works. Long-time listener. Love what you do. You're an inspiration for all science communicators. Thank you so much, Dr. McPharma. I appreciate that very much. I'm glad what we do, because it's a collective effort, I'm glad what we do has an effect. The movie Andromeda strain is a non-living infectious agent. Are you sure? I thought it was a bacterium. Shaking and quaking. So could we put prion-infected people into ultrasonicators? No. It's just a test for detection, right? We don't want to put the people in it. Thank you very much. And thank you for your heart and thumbs up. I'm glad that you all came tonight to listen. And let me thank our moderators tonight. We had Steph. We had Les. We had Tom. We had briefly Vanity Nutrition. We had briefly, I forgot his name. Who's the first guy that was here in the very beginning? No, Tom. You can't go all the way back. Peak was here. Thank you all for moderating once again. Appreciate your service, and thanks all of you for coming, all 200 plus of you. You have great questions. Next time was going to be next week. Let's see. Next Wednesday is the 15th of... I'm going to be in Princeton for the textbook, so I don't think I can do a live stream. And so I will have to let you know. I'm doing some traveling for the next few weeks. So go check out the website, Microbe TV, for the schedule and see when the next live stream, because it's not going to be next week because I'm in Princeton working on the textbook. We'll try and see when I can do it again. But anyway, folks, until whenever it is that I see you, please, thank you for coming and be safe. Good night, everyone.